My talk will be on quantification in optical imaging, just really aiming to give you the basics of the physics involved in optical imaging and a few different techniques that are used and being developed. Those are the contents. I'll start by describing interactions between light and tissue so that you have the basic understanding, and then I'll go on to describe three different techniques: simple planar imaging, fluorescence molecular tomography or FMT, and multispectral optoacoustic tomography, so that you have an overview of what you can do with each method. Optical imaging, in particular using fluorescence, is quite well established in biomedical research in the microscopic domain, fluorescence microscopy, etc. But more recently, there's been a lot of interest in expanding that to macroscopic imaging of larger fields of view and deeper tissues. But unfortunately, that brings a whole lot more complexity in terms of quantification, because instead of looking at just one thin slice where you can see exactly where your signal is coming from, you have a volume and some effects that are very difficult to model. The reason it is pursued, however, is because optical imaging has a number of attractive properties. For one thing, the contrast is quite powerful, with contrast sources ranging from bioluminescence and fluorescent proteins for gene expression, visualization, and a number of molecular fluorescent agents. Optical imaging is multispectral, so you can use different wavelengths simultaneously to view different biological targets, and it's non-ionizing, unlike X-ray based or radioisotope based imaging. It's quite safe to use and also relatively economical in terms of instrumentation. Two interactions of light with tissue really hinder the propagation of light, and the first one is absorption. And you can see over there I've plotted the absorption of light for visible and near-infrared wavelengths in average tissue, and it's actually dominated by absorption by hemoglobin. And what you can see, which is really the key point, is that in the near-infrared wavelengths, in the longer wavelengths, there's orders of magnitude less absorption of light in tissue than there is at visible wavelengths. So while green light, for example, will only really illuminate the skin or the tissue surface, red light or near-infrared light can pass through several centimeters. And you see that demonstrated at the bottom of the slide on those two images. Green light shining through a mouse, you don't get much signal, whereas if you shine red light at 670 nanometers, it really penetrates through several centimeters and gives us a useful sort of window in which to image. Absorption can be described in terms of an absorption coefficient. That's a property of the tissue, mu A over there, and that describes really the decay rate of the intensity of illumination in a purely absorbing medium as light passes through it. So that's absorption described by mu A. Unfortunately, that's not the only interaction that causes problems in optical imaging. We also have scattering, and scattering is the deviation of light away from the direction in which it was traveling. So if you look at that photograph, that's a beam of light traveling through a glass of water. And if we add some drops of lipid to that water, suddenly the light is scattered, it becomes diffuse, and we really have difficulties telling where that light is coming from if we look at one particular point. And that's really the situation in tissue. Tissue is very highly scattering. Scattering can be described by a scattering coefficient, mu S, and that gives you, at least if you take the inverse of that, that gives you the mean distance between two different scattering events. So how far a photon on average can travel before it's scattered. Scattering in tissue is not isotropic generally. It's very forward scattering, so the photon's actually scattered around the direction that they're traveling in preferentially. And that's defined or described by an isotropy number G. The closer it is to 1, the more forward scattering the tissue is. In highly scattering media, and tissue is generally highly scattering, this whole situation can be approximated after the first millimeter or so by isotropic scattering with what is called a reduced scattering coefficient, mu s prime, and mu s prime is just 1 minus g multiplied by mu s, and that gives you a value for scattering, assuming that in fact photons are scattered in all directions in a very diffusive situation. And as you can see from those values in the table, reduced scattering coefficients in tissue are usually in the range between, say, 10 and 30 inverted centimeters. And it's scattering that really causes the biggest problem in terms of reconstruction of optical imaging and optical imaging in general. If I compare it here to X-ray CT reconstruction, where scattering to a good approximation does not occur and X-rays travel in straight lines, we then only have to consider the straight line path between sources and detectors for reconstruction. On the other hand, in optical imaging, as you can see in that image on the bottom right of the slide, photons traveling from a source at the bottom indicated by S to a detector can essentially travel through any pixel or voxel in the volume. And so this whole sort of distribution in that image has to be considered in the image reconstruction algorithm, and it's very difficult to tell where signal is coming from when photons could be traveling through any one of those paths. The contrast mechanism that I'll be focusing on for most of this talk is fluorescence. Fluorescence is essentially the emission of longer wavelength or lower energy photons after the absorption of higher energy or shorter wavelength photons. And the reason this happens is that a portion of the absorbed energy is given off in vibrational relaxation faster than a photon is emitted. So you end up with a situation in a fluorophore. There's a plot of Psi55, a common fluorophore over there. You end up with a difference between the absorption maximum at a lower or shorter wavelength and the fluorescence emission maximum at a longer wavelength. And this enables you to actually use an optical filter to separate the excitation light from the emission light and have very sensitive detection of the fluorescence emission. And that's what makes optical imaging or fluorescence imaging so sensitive in the first place. Fluorescence can be used in a number of different ways for molecular imaging. For one, a simple way is fluorophore tagged selective targeting, where some receptors, say, are targeted by something that has a fluorescent tag on it. Another method is activatable agents, which by some dequenching mechanism only start fluorescing when they are, for example, cleaved by certain proteases. And of course you have a wide variety of applications for fluorescent proteins, such as visualizing gene expression and cell labeling, etc. So the imaging aim, once you have fluorescence in your system, is to determine the distribution of the fluorophore in the tissue volume. And The simplest approach to do that is what we sometimes call planar imaging, and it's essentially a matter of taking photographs of the tissue. You have illumination to excite the fluorophore, and then some sort of filter to filter out the excitation light and leave you only with the emitted fluorescence and a camera to take the images. Now, in the case of bioluminescence, of course, you wouldn't have the illumination or the filter, you'd just have a dark room or dark box. So the situation is very similar. What can we say about quantification in this case? Well, quantification is hardly possible. It's hindered by absorption and scattering, because any light, whether it's the excitation light reaching the fluorophore or the emitted fluorescence light, is going to be scattered as it travels through the tissue. And this means that signal from the surface, so for example on the skin or very close to it, will really dominate compared to signals from deeper inside the tissue. Another problem that we could face with quantification is variations or heterogeneities in the optical absorption properties of the tissue being imaged. And there's an example of that on this slide. Those images show two mice. The images in the left column are under excitation light, so without a fluorescence filter, and the images on the right are the fluorescence images. And these mice have tumors that have been targeted by some fluorescent agent. And you can see in the top row that there are two tumors on the breast which are lighting up as we would expect. But in the bottom row on that mouse, the tumor which you can see again on the breast is very dark in the excitation image, and that means it's very absorbing. It's absorbing a lot of light, probably contains a lot of blood, and as a result, the fluorescence image is distorted by the fact that the fluorescence emission light is being absorbed and is unable to reach the camera to the extent where it would give any contrast over the background. There are methods to somewhat alleviate these problems. One of them is to normalize the fluorescence image by dividing it by the excitation light image, and this slide shows two examples of that. In the one case, using tubes that have the same concentration of fluorescence or fluorophore, but increasing amounts of absorption, and if we then normalize those images, we recover more accurately this sort of constant level of fluorescence. And in the case of those ex vivo images with lymph nodes, again, one of the lymph nodes is very absorbing. It causes the fluorescence image uncorrected on the bottom left there to be incorrect or to give a skewed picture, and the normalization somewhat solves that problem. So far we've looked at imaging in the reflectance mode or epi-illumination, where the illumination is on the same side of the tissue as the camera. What can also be done to alleviate the surface weightedness of planar imaging is to actually put the illumination on the opposite side of the tissue, as shown on the right-hand side there. And this will, to a certain extent, improve the surface-weighted problem or solve it. And this slide shows simulations intended to clarify that. So on the left-hand side, the situation shown is bioluminescence. You have two sources indicated by the sort of red circles. And if you image this situation from the top, the source closest to the top will dominate. If you image from the bottom, the source closest to the bottom will dominate the signal. So you're only ever seeing the signal source that is closest to the tissue surface, because the other signal, the other light, is being attenuated by scattering and absorption. In the case of epi-illumination or reflectance mode imaging, it's exactly the same. You see the source that's closest to the side that you're illuminating. However, in transillumination mode, where the illumination is on the opposite side, all the light has to pass through the entire tissue volume or the entire tissue thickness, and so you have comparable signal levels for sources at different depths, and you have less of this surface weightedness. The next sort of step in quantification is to use a tomographic approach, for example, fluorescence molecular tomography or FMT. And in this approach, a point source of light is essentially scanned over the surface of the tissue or the small animal being imaged, and images from the opposite side, so transillumination images, are captured for each of these scanned point sources and for a number of different rotational projections. So the input is the scanned point source of illumination, and the output are these sort of planar images in transillumination mode. FMT uses normalized data. Again, fluorescence images like that on the bottom left are divided by images of the excitation light, shown in the middle there, to give you a normalized fluorescence image. And these images are fed in to a reconstruction algorithm that then processes them further. FMT image reconstruction is model-based. As I pointed out, because of scattering, you can't use very simple back projection like you would use in CT imaging. So we have a model-based approach. A forward model describes how light propagates through the tissue, and this is formulated in a matrix together with the normalized measurement data. This is then inverted to gain the fluorophore distribution in the volume. And because of optical scattering, this is an ill-posed problem, so there are limitations on the resolution that you can get in this fluorophore distribution. The forward model is based on the diffusion approximation to light propagation, so we assume that scattering is isotropic for one thing. And there are essentially two diffusion equations. The top one describes how the excitation light propagates from point sources through the tissue, and the bottom equation describes how the fluorescence emitted light, which comes from a combination of the fluorophore distribution and the excitation light, propagates towards the detectors or through the tissue. A Green's function approach is used then to describe the solutions to these diffusion equations, and that really tells you what possible paths light takes between point sources and the point at which you're detecting, or points of interest. So, for example, in that diagram, between a point source and a point detector, light could take this sort of banana-shaped distribution to get there. So this slide just gives an example of some older standalone FMT results, because I think it illustrates the kind of capabilities you have with FMT over and above planar imaging. This is a mouse model of myocardial infarction injected with magnetofluorescent nanoparticles, and they get taken up by macrophages in the infarct. And you can see over there those images are essentially white light photographs of mice superimposed with the fluorescence distribution in a coronal slice through the heart. And the image on the left is an infarct mouse, and the image on the right is a control. And there were actually quantities taken out of these images. So it would otherwise not be possible just using single planar images to actually take something as deep-seated as the heart and compare quantitatively amounts of fluorescence in controls to the infarct animals. So this really demonstrates why you would want to use a tomographic approach in the first place. Of course, it's very difficult in those images to see where the fluorescence signal is coming from because there's no anatomical information that comes with it. It's really just the distribution of the fluorophore. And so it makes a lot of sense, and it is quite common these days to use FMT together with another modality, for example, X-ray CT, as shown over there, to give you anatomical information combined with molecular imaging. And that gives you not only this co-registered combination, but it also allows you to use the CT information, or MRI in some cases, to actually improve the image reconstruction obtainable in FMT. So as one example, you could take the CT volume, segment it into different organs or tissue regions or tissue types, and then assign more accurate optical properties to those regions to make the forward model in the FMT more accurate and therefore obtain more accurate results. So this is something that is actually done. This is a picture showing the first hybrid FMT-CT imager for small animals. It's essentially a micro CT with FMT cameras and lasers and filters added to it, and this system rotates around 360 degrees to get all the projections necessary for good FMT reconstruction. These are the kinds of images you can get from such a system. This is a mouse model of Osteogenesis imperfecta injected with an osteogenesis targeted probe, and the fluorescence distribution is shown there in this sort of golden color, and it goes to the spine, as we can see if we zoom in, and also if we zoom in further we can see that it corresponds to regions of bone remodeling. So this is something we absolutely could not do if we were just taking single images, and it would also be extremely difficult to tell where the signal is coming from if it weren't combined with CT so that we could see the anatomy of these animals. As I mentioned, optical imaging is widespread in microscopy, and within the first few hundred microns from the illuminated tissue surface, we can actually get extremely high resolution images limited by optical diffraction. However, as soon as light starts scattering, which is after a few hundred microns, then the resolution is really degraded by this sort of blurring of the light. And no matter how sophisticated our reconstruction algorithms become, there's no way of getting around this. So FMT gives you a spatial resolution of about 1 millimeter after approximately 1 centimeter of tissue penetration, which are about the dimensions of a mouse, at least to the center of a mouse. And obviously, any way to sort of overcome this scattering barrier would be quite attractive. And that's one of the motivations behind optoacoustic imaging, which is a way really of getting around the barrier that the scattering of light puts up to high resolution. Optoacoustic imaging relies on short pulses of light that illuminate the tissue. That light then, after it's been scattered, it doesn't matter what happens to the light, it is absorbed in the tissue by certain absorbers. Those absorbers undergo thermal expansion, and that gives rise to broadband pressure or ultrasound waves that can be then detected non-invasively. Now, if we detect those waves at multiple different spatial positions, we can reconstruct an image of where that light was absorbed or the initial pressure distribution. And as you can see on that formula there, that is actually proportional to the optical absorption coefficient. So we can see how absorbing or how much light is absorbed spatially in the tissue. And the reason that it is not limited in terms of resolution by optical scattering is because we're detecting this by means of ultrasound. And ultrasound scatters orders of magnitude less than light in tissue, so we can maintain quite high resolutions defined by ultrasound even after several millimeters or centimeters of tissue penetration. Multispectral optoacoustic tomography, or MSOT, is really the multi-wavelength extension of optoacoustic imaging. I've plotted there the absorption spectra of oxy and deoxyhemoglobin, which dominate tissue absorption in most scenarios at these near-infrared wavelengths. And we can add an exogenous absorber, for example, indocyanine green or ICG, and then we image this tissue at a number of different wavelengths optoacoustically and use spectral and mixing techniques to then separate the images for each of these characteristic spectra. And so what we end up with are specific images of the intrinsic absorbers, such as hemoglobin in its different oxygenation states, and also any sort of optically absorbing agents that we have added to the tissue. Just to give you an overview of what sort of contrast we can get from MSOT imaging, this slide shows some tumor imaging. There's a subcutaneous tumor there on the back of the mouse, and the white dotted line shows the imaging plane. And that tumor, or the tumors, show up then at the bottom of the optoacoustic images that are shown on that slide. In the case of the animation or the video, this is a mouse being injected with indocyanine green, the near-infrared dye, and as it's injected intravenously, you can see this green signal, which is the contrast enhancement from indocyanine green entering the vasculature in and around the tumor. And this is looping, so you can see it from the beginning again. This is essentially real-time imaging, and the green is just showing the contrast enhancement over the baseline. In contrast to that, the image on the top right shows the same situation, but instead of imaging over time, this is imaged at a single time point multispectrally and spectrally unmixed then to find this ICG distribution, again indicated in green. So you can see those results agree with each other quite well, and without the need for a baseline measurement, you can spectrally identify the different chromophores in the tissue. A further chromophore or chromophores that you could identify are hemoglobin in its different oxygenation states, and that's shown on the bottom of the slide. This tumor was imaged over several days, and from that multispectral data set, you can extract the distributions of oxygenated and deoxygenated hemoglobin, as is shown there. And you can see the viable vasculature on the periphery of the tumor and this core of deoxygenated blood in the middle there. So these images of this tumor that has a diameter of about 5 millimeters, I think, have a spatial resolution of about 150 microns, and optically there's no other way to penetrate that kind of depth, several millimeters, but still retain this kind of resolution. FMT would give a lower resolution, planar images would give surface-weighted images, so this is quite unique in its capabilities. To summarize now, this table just shows some of the performance characteristics that I've spoken about of the different techniques that were introduced. from the most simple planar fluorescence imaging. The contrast is based on fluorescence. It doesn't really have any quantitative accuracy at all, so in fact it's mainly useful for quickly determining whether something is fluorescing or not, as opposed to trying to get quantification out of it. It has an undefined spatial resolution because what you're seeing is essentially the surface of the image, and anything that is coming from below the surface is highly scattered depending on depth. But on the other hand, it's very simple. It has a high temporal resolution and can in fact be used in video mode for real-time applications. Fluorescence molecular tomography is of course also based on fluorescence as a contrast mechanism. It brings quantitative accuracy to fluorescence imaging in deep tissues. It has a spatial resolution limited by scattering of about 1 millimeter in objects the size of mice, and a temporal resolution which varies according to system, but is commonly measured in tens of minutes because each projection has to be obtained sequentially. So of course this isn't something for imaging dynamics over very short periods of time. Multispectral optoacoustic tomography, on the other hand, is based on absorption contrast. So it doesn't measure fluorescence, but how much an agent or a chromophore absorbs light. It gives quantification based essentially on spectroscopy by measuring absorption at different wavelengths, and has a spatial resolution that is defined by the ultrasound frequency, but for objects the size of mice could be around 150 microns, and has a high temporal resolution because we can use ultrasound detector arrays to measure signals in parallel. So that's really all I wanted to say about optical imaging. I hope that it's been useful just for a little bit of deeper understanding of where quantities come from and what they depend on. Thank you very much.